Hello everyone, it's Jobs here, and in today's video we are going to learn about the metabolic pathway known as glycolysis. So what is glycolysis? Glyco refers to sugar, and lysis means breakdown. So you can already assume that glycolysis is the metabolic pathway involved in the breakdown of sugar. Before we start, I would like to ask you to leave a thumbs up for this video and subscribe to my channel. This will help me improve my content. Now let's get straight into it. Let's assume you purchase a candy, and while you're chewing it you wonder, how does this turn into energy? The candy you just ate can be represented as a big chunk of sugar molecule complex in form. As soon as you start chewing the candy, the enzymes within your saliva, specifically amylase, start breaking down the complicated sugar structure, aiming to produce a more simplified and digestible form, known as glucose. In brief, the breakdown process is carried until glucose is produced and has reached your intestine. Here it will be absorbed into the bloodstream. Your blood will be responsible of delivering glucose to cells within your body, and here, finally, glycolysis can take place. So what is glycolysis used for? This metabolic process is used to convert one molecule of glucose into two molecules of pyruvate. Glycolysis can be broken down into two major categories: energy investment phases, where energy is consumed as you land your energy, aiming to have a profit in return over time. Then there are energy payoff phases, where energy is gained and the profit on the initial investment is made. Let's now explore each step. To begin with, the first 5 steps cover the energy investment phases. Step 1 is known as phosphorylation. The glucose molecule entering the cell gets immediately phosphorylated, and here energy is consumed in the form of ATP. The phosphate will be stripped off from ATP and gifted to the 6th carbon within the glucose molecule, reason why the product has a nomenclature of glucose 6-phosphate. The enzymes involved in the catalysis of the reaction is hexokinase if the process occurs in the brain or muscle tissue. Instead, if the process occurs in the liver, the catalysis will be done by glucokinase. The phosphorylation process is a crucial step to trap glucose inside of the cell. If phosphorylation does not take place, the glucose molecule will leave the cell and glycolysis will stop. There are disorders associated with this particular step not occurring during glycolysis. Now let's move on to the second step of glycolysis, known as isomerization, which simply refers to the rearrangement of the glucose molecule structure. Here the process is catalyzed by phosphoglucoisomerase, which converts glucose into a fructose molecule, reason why fructose 6-phosphate is produced. In step 3, another phosphate group is added, and energy again is consumed. This phosphorylation prepares the molecule for cleavage. The phosphate stripped off from ATP will produce fructose 1,6-bisphosphate. An easy way to remember this molecule is that phosphorylation in step 3 adds the phosphate on carbon 1, and because of that, now the molecule has 2 phosphate groups, represented in the nomenclature by the prefix bis. This process is catalyzed by phosphofructokinase. Step 4 can be described as splitting. Here, the sugar molecule is split into 2 molecules by the enzyme known as aldolase. This forms dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Here, it is important to know that only glyceraldehyde 3-phosphate is suitable for the next steps of glycolysis, and dihydroxyacetone phosphate isn't. Reason why the next step of glycolysis is fundamental in order to modify dihydroxyacetone phosphate. This involves another isomerization step. Triose phosphate isomerase will catalyze the reaction. Triose refers to 3 as the number of carbon atoms in dihydroxyacetone phosphate, which will be converted into another 3 carbon atom molecule, again glyceraldehyde 3-phosphate. After the conversion, it is now understandable that we are dealing with 2 molecules of glyceraldehyde 3-phosphate, continuing the steps of glycolysis. Now, let's pause for a second. If you feel lost or overwhelmed, take a small break, get a coffee or whatever you fancy, watch the video again up to this point or from where you feel you have lost the track. It is more efficient to build your knowledge piece by piece instead of throwing all at once. Now let's resume. The following 5 steps are the ones involved in the production of energy. It is important to note that we are dealing with 2 molecules of glyceraldehyde 3-phosphate, and because of that, every product obtained during the reactions will account for 2. The 6th step involves a redox reaction and a phosphorylation. The redox reaction happens when NAD+ removes a hydrogen from glyceraldehyde 3-phosphate and produces NADH. NADH is another form of energy, thus the first currency of energy is produced. Then the inorganic phosphate will be used to phosphorylate carbon number 1 in glyceraldehyde 3-phosphate, and this will lead to the production of 1,3-bisphosphoglycerate. Both reactions are catalyzed by glyceraldehyde phosphate dehydrogenase. Dehydrogenase simply refers to the removal of hydrogen from glyceraldehyde 3-phosphate by NAD+. In step 7, the first ATP is formed. ADP removes the phosphate from carbon 1 in 1,3-bisphosphoglycerate and produces ATP. This occurs through the catalysis of phosphoglycerokinase for the production of 3-phosphoglycerate. In the next step, the phosphate gets relocated to a different position within the molecule. In fact, phosphoglyceromutase, which is different from isomerase, moves the phosphate group from carbon 3 to carbon 2. This forms 2-phosphoglycerate. The next step settles the foundation for pyruvate formation. This occurs through the catalysis of enolase and produces 2-phosphoenol pyruvate. Finally, the last step of glycolysis forms pyruvate and ATP. Here, ADP will strip off the phosphate from 2-phosphoenol pyruvate and will form ATP and pyruvate. This reaction will occur through the catalysis of pyruvate kinase. Remember, every product seen so far are doubled, as a process initiated with 2 molecules of glyceraldehyde 3-phosphate. So what is the net income from the initial investment? The net income accounts for 2 NADH, 2 ATP, and finally 2 pyruvates, which are going to be utilized by the Krebs cycle to produce more energy. I hope you enjoyed this video. If you did, leave a thumbs up and subscribe to this channel. Thanks for watching and see you in the next video.